hand over to Ammasi. The title of his talk is Investigation of Metabolism in Living Prostate Cancer Cells, Two-Photon FLIRR Microscopy. Ammasi, please start.
Thank you, Hake, and thank you introduction for the Wolfgang. So I am going to talk about the investigation of metabolism in living prostate cancer cells, two-photon FLIRR microscopy. So before that, the acknowledgements goes to a number of people involved in the project. You can see, oops, can I move it? Yes. Yeah, so the pointer disappeared. 1 second. Yeah, so Sagupta and Zdenek, host Rabi, and here listed Rufon, and Carsten Siller is mainly involved in the image segmentation. Daniel Weller is mainly involved in the machine learning. We are just introducing machine learning, how to screen the Jiashin is a student. Yang is also involved in the project. I will show you some of the data. It's frozen again. I am not sure what's going on. Sorry about it. It's
Ammasi,
frozen again.
Ammasi, can you please switch off the this blur effect from Zoom, that you see the mouse point, this mouse pointer, because I think this makes everything slower.
Okay, now?
Yeah, this is probably better. Try again. Does it? Huh.
It's giving trouble. 1 second. It was working okay. Okay, all right. So you should share the authorship. That's what I am trying to show. And the funding is from different organizations, which is NIBIB, Office of the Director, Institute of Aging. Recently, we received an award from Institute on Aging, Cancer Center, and University of Virginia. As I mentioned, that I am going to talk about how to investigate the mitochondrial dysfunction using lifetime imaging microscopy and also FLIRR, phosphorescence energy transfer. As you know, the cells has the mitochondria. Most of the cells in our system, you see that the mitochondria, where the mitochondria is the energy source of the cell survival, and it converts the ADP to ATP. You can see clearly it goes to all over our body organs. But only the blood cell doesn't have the mitochondria, but all other cells has the mitochondria, which we are investigating. There are a number of works is going on the metabolism work. So here I am mainly focusing on NADH measurements, and glucose is degraded to pyruvate upon reduction of NAD plus to NADH. Pyruvate is preferably converted to acetylcholine and fed into the TCA cycle in the mitochondria. The proton gradient built up over the inner mitochondrial membrane is used by complex 5 to convert ADP to ATP. So that is the ATP is the energy source for the cells. So I am going to talk about mainly on the NADH measurement for drug response in prostate cancer cells, and as well as for the leukemia cancer cells, and how we developed a new idea of implementing how to do that data analysis. As you know, the cancer now currently decreasing, the death rate is decreasing. Probably it is due to the development of the diagnostic process and also the technology advancement for the treatment. So if you look at the prostate cancer, it's about, they expect, CDC, Center for Disease Control, expects that there will be a decrease still further if you see the graph. On the y-axis, the death rate, and the x-axis is by year. You can see the decrease in death rate. Probably it is due to the advance in technology and the diagnosis and treatment. So treatment and diagnosis of prostate cancer is a much debated subject. If you walk into the doctor's office, immediately they will ask you, you want to have a PSA test, that is prostate specific antigen. It is still important indicator, but PSA test is implemented only, it will show up only if you are a positively diagnosed patients. We are trying to see whether we can do early detection of prostate cancer using this metabolism or endogenous molecule, monitoring the signal using the lifetime microscopy method. To investigate it also, we are, as I mentioned, investigate the cancer drug response for the prostate cancer cell, and we also work on the metabolic FLIM approach, So the recent evidence has suggested that cancer should be considered as a metabolic disease. Growing tumors rewire their metabolic programs to meet, even exceed, bioenergetic, biosynthetic demands of continuous cell growth. The metabolic profile is observed in cancer cells, often includes increased consumption of glucose, glutamine, and increased glycolysis, changes in the use of metabolic isoforms. So why do cancer cells shift their metabolism? Are the changes in metabolism in cancer cells a consequence of the changes in proliferation or a driver of cancer progression? Can cancer metabolism be targeted to benefit patients? So these are the recent questions are raised in the metabolism work. So there are a number of methods are used to monitor or measure the cellular metabolism. Radioactivity or pH measurement, mass spectrometry, or NMR spectrometry. We are using fluorescence microscopy techniques, which provides spatial and temporal information of the drug response from the cells. So terminology I may be using, most of you know that if it is a FLIM group, you will be familiar with all this terminology I am going to use. Mitochondria, that is what we are investigating. The coenzymes, NADH or NADPH, and FAD, flavin, and amino acid also we are investigating on the tryptophan side. The cells we will be using prostate cancer cells and acute myeloid leukemia cells. The drug used for the investigation is doxorubicin for the prostate cancer cell and ceramide nanoliposomes for the leukemia cancer cells. The technique which we use is the two-photon fluorescence microscopy and two-photon fluorescence lifetime imaging microscopy. And the technique newly we developed the fluorescence lifetime redox ratio, which we call it as a FLIRR. Of course, the data analysis, I will show you how to do that. So the tryptophan absorption is 295. You cannot use the UV, so we are using 3-photon excitation, 740 nanometer, and for NADH, 2-photon excitation, 740 nanometer. The emission is in the UV for tryptophan. The microscope is configured for the UV emission from 300 to 1200 nanometer. This is the size 780 scope. You can see 3 PMTs simultaneously at the same focal plane. We can collect tryptophan, NADH, and FAD. These are all systems from Becker and Hickel. And FAD excitation, we are using 890 nanometer. All the data given here, two wavelength excitation. As you see, if you excite with the 740 nanometer, the mitochondria is in the cytoplasmic area. You can see the green color represents the NADH signal. We label the mitochondria with a mito tracker. You can see red color when you co-localize. You can see what we are collecting signal or from the mitochondria, all the NADH signal we acquire from the mitochondria. So fluorescence lifetime imaging microscopy, many of you know, I don't have to give deep in the discussion, but I would like to cover a little bit. As you know, what we are measuring lifetime is if we excite or illuminate the light source, any fluorophore molecule, it goes to the excited state and return back to the ground state. We are measuring average lifetime, how long the molecule stays in the excited state. The most of the fluorophore molecule we use for biological application within 1 to 5 nanoseconds, even microseconds, some of the fluorophores, depending on the solvent. Mainly the lifetime depends on the solvent effect. If you change the solvents, dissolve the fluorophore, the lifetime value will change. So mainly it's sensitive to environmental changes of the biological process, insensitive to change in molecule concentration, or power of the excitation intensity, or light scattering, and to some extent photobleaching. So the FLIM microscopy technique provides spatial and temporal information of the biological events. There are two methods in lifetime imaging, as you know, frequency domain and time domain. And what I am going to present is mainly time domain, single photon counting. And lifetime data analysis, we use the Becker and Gickel software to non-linear least square fitting the data, or we can use the phasor method and then use the data, resultant data after fitting. It provides number of parameters, maybe 30, chi-squared value, a pre-exponential factor, A1, A2, and number of tau 1, tau 2 is most of the time for NADH, we fit for 2 components. Analysis, same as the tryptophan, we fit for 2 component analysis. So using the data, we developed image segmentation, and then using the number of data, we use the machine learning to how to extract the information from the collected data. So how I got into this lifetime imaging in 1990s, there is no lifetime imaging system at that time, like now you can buy from any companies and use it in your lab. At that time, it was available only in the physics or chemistry lab where they know how to build the system. So I was working on calcium imaging and calcium, using calcium crimson fibroblast cells. This is what we found out the sensitivity. You can see increase in signal in the nuclear area. When we submitted the paper, the reviewer said, no, this is not right. Biologically, it is wrong. So then that is where we developed the lifetime imaging system. Clearly it shows that there is no calcium binding in this experiment. So it's a very narrow peak. So that's how I got involved in the lifetime imaging system, developing the system. Here it is. You can see all the electronics you have to put together if you know how to build the system. And with all high-end laser system, since you are measuring the nanosecond lifetime, you need the detector, high-speed detector to do that. All right. So now you can, all the single photon counting electronics in a single board, you can easily buy it and put it in your PC, connect to the scanning microscopy system. You can do your lifetime, you can collect lifetime imaging using a confocal or two-photon microscopy system. So we wrote a review article on investigating protein-protein interaction in living cells using in the Nature protocol, where we wrote plus and minuses of the techniques, so you can step-by-step instructions using Becker and Gickel software instruction, and also ISS, frequency domain method. And also book, Wolfgang also edited a book, and myself and Robert Klug, we edited a book on the lifetime imaging microscopy. All right, so a brief review, you excite the laser pulse, so you get your fluorescence decay. Then during the decay, you count the photons, and each pulse gives you a fluorescence decay, and then you count the photons, and the resultant will be, you can see a fluorescence decay. So if from this decay, you do not know whether it is a single component or 2 components or 3 components is available. So you have to fit and see. But it will not tell you where the signal is coming from. in the cells is nucleus or cytoplasmic. That is why the imaging method came in the picture. Using the microscopy techniques, you can able to see the distribution of the lifetime distribution from a cell. So in here, the NADH provides free is 0.4 to 0.7 nanoseconds, and the bound NADH 2.4 to 4.0 nanoseconds. So this kind of discrimination of free and bound is possible to do that only using a lifetime. Using the intensity method, it's very difficult to separate the free and bound NADH signal using a spectrophotometry. So that is why the lifetime technique is advantageous in working with discriminating different information of the cellular biology is complex. So that is the cellular information. So here is, if you use a fluorophore alone, you get a single component. And if you just throw the fluorophore in cells, you can see a distribution. If you target to the cells, again, you see a 2 components or 3 component analysis. It may be due to solvent effects or something biological events. So if you use a FLIRR, you can see a single component, 1 fluorophore donor alone. If you use a FLIRR donor plus acceptor, say for example, cerulean and venous, you will get 2 exponential decay, 2 lifetime distributions of the donor. So how do you do the analysis? That's where, after fitting the data, as I mentioned before, we developed a software image segmentation approach using region of interest selection. And then we use the 2 parameters, the bound NADH and the bound FAD ratio will give you the redox ratio, which provides the drug response. And now recently we are introducing the machine learning approach. Okay, the lifetime gives a number of parameters. Those are all contribution to the lifetime distribution of the molecule, whether it is free or bound. So we thought that we should use the machine learning to use other parameters involved in the contribution, whether we can improve the detection sensitivity compared to the FLIRR value or the intensity-based redox ratio. So conventionally, it is available. Many literature shows you that the redox ratio is calculated by intensity, ratioing NADH bound non-fluorescence, the NADH signal over the FAD signal. So what we used is using this lifetime approach, we use the enzyme bound NADH and also the enzyme bound FAD signal. If you fit the data, you can see you will get A1%, A2%. We just directly pick up from the data of using the SPC image from Becker and Gickel, and then we ratio those image after segmentation. But this approach, it's nothing wrong in the intensity-based approach. This approach is very well avoid all the intensity artifacts involved, particularly in tissue in live animal. When we did the live animal work, that is where we found out that it is an issue with intensity-based ratio. I will show you. So here is an example. It's a live animal work. You can see tumor versus cancer. It's all over the place. It's supposed to give you uniform distributions with our testing. So it's maybe due to the intensity light scattering in the tissue and all kinds of differential absorption of light in the tissue. It gives this kind of information. Using our FLIRR method, this bound NADH divided by FAD bound coenzymes, it gives you a clear, not jumping around, or the data is very reproducible. So you can see that from the cell and as well as from the tissue, the pattern remains the same, the NADH and FAD signal with the cancer versus normal tissue. So now I'll show you the cellular metabolism, how we are processing the data. People process the data with excluding the nucleus, taking the whole cell. But the ROI method, which we introduced, is provide you to also differentiate. Not only this image segmentation method goes and select the mitochondrial area in the cytoplasmic, it gives you the separation of the OXPHOS and glycolysis. Glycolysis in cytoplasm, OXPHOS takes place in the mitochondria. So this software allows you to discriminate both things, and also it gives you, allows you to avoid any intensity artifact involved in the microscopy imaging. So here you can see whole cell versus mito alone. Here we grouped into 3 categories, lower and minimum in the middle. You can see the distribution, the heterogeneous distribution of the whole cell versus the mitochondria alone. You can see clear separation in the mito alone if instead of taking whole cell, which includes glycolysis and the OXPHOS information too. So how do you test? What we are doing is our approach, the FLIRR approach is right. As you know, the Gruff cycle, the blocks, the cobalt chloride blocks the connection between the Gruff cycle and OXPHOS. So that if you use the cobalt chloride, then the production of NADH decreases. So if you add the glucose, then it should increase. So that way we can test whether our FLIRR approach is working or not. If you see the cobalt chloride here, you keep the cobalt chloride about 50 micromolar, pre-treated overnight, and imaged as a control. Then challenge with 25 millimolar glucose for about 30 minutes. Then image it and then try to process our region of interest-based method. So we can see the intensity-based ratio here and the FLIM ratio here. And the bottom panel is FLIM ratio, and the top panel is intensity-based ratio. You can clearly see the arrow mark indicate the green, the increase in sensitivity. The pattern remains the same, both intensity and the FLIRR ratio, but the sensitivity increases using the FLIRR ratio. Probably it may be due to the to reduce the intensity artifacts. All right, then we used the LINCAP and African American cancer cell line to see the drug toxorubicin. You can see 15 minutes, 30 minutes, and 45 minutes, and 60 minutes. So you can see the drug response. The black color, this first one represents the control, and then the other color pattern shows the increase in redox ratio with the time with the toxorubicin. And after a while, you can see 60 minutes, the morphology of the cell changes with the toxorubicin. I wanted to bring that up later. The African American cell line, prostate cancer cells, not that much resistance to, it always resistance to the drug compared to the LINCAP cells. It may require more concentration of the drug to respond well. So this is the enzyme bound response of the LINCAP cells and the African American cancer cells. We just measured the, the FLIRR value is the bound coenzymes of the NADH. And this is the FAD, bound FAD decreases with the drug with the time. You can clearly see the control is here, the block line, and the response is from in the other side, this is decreases 1%. Both LINCAP and African American prostate cancer cells. So we also tested the biopsy, prostate cancer biopsy tissue, which also demonstrates that the intensity based on the right side, it jumps all over the place, whereas the FLIRR method is, provides very stable, reproducible output. So the redox ratio calculation using bound NADH divided by FAD, bound FAD, is really gives you a positive or reproducible results. So why do you want to do this kind of calibration and verification? If you want to approach a doctor to get biological samples, you have to demonstrate that this system, this configuration is works. So we tested with the clinical samples. This is the clinical samples from different blood samples from different patients, and we were able to measure the control is orange color and cancer is the blue color. So we were able to measure the system. It is the reproducible with the PSA. We are comparing with the PSA. It's reproducible about 4% to 5%. So we need to continue this work. Okay, this ends with the demonstration of our explanation of the prostate cancer cell testing with FLIRR or REDOX ratio measurement to calibrate or to measure, monitor the REDOX ratio of the cancer cell for toxorubicin drug. Okay, so now I would like to show you some data FLIRR between the tryptophan and bound NADH. Tryptophan lifetime quenches in the presence of bound NADH, which we tested already, those things. So before that, I think most of you know about FLIRR. To flow up the talk, I will also tell about the basics of FLIRR a little bit. As you know, most of you know, we are running the workshop for the last 19 years. And I am not sure next year we will have the hands-on training workshop because of the virus situation, or we are not sure still, we are not decided to go for virtual because the purpose of the workshop will not be there if you go for virtual because it's a hands-on training course. Probably we will postpone for 2022. We have not decided yet. Faster resonance energy transfer provides you about dynamic behavior of the biological molecules. It's basically a communication between two fluorophore molecules or two protein molecules. It provides you live specimens you can use to measure the FLIRR. So you can do single cell assay, investigate subcellular compartments, and spatial and temporal information of the interacting proteins. So what is FLIRR? FLIRR is a process, many of you know, radiation and transfer of energy from one fluorophore from the excited state consideration to an acceptor in the ground state in close proximity. So it will be a radiationless energy transfer from one molecule from the excited state to the ground state to the acceptor. If you use the lifetime technique, the donor lifetime will decrease if the conditions are satisfied, meaning that the distance and the dipole moment, two conditions in addition to spectral overlap. So there are three conditions, main three conditions. There are other optimization you can do. One, the fluorophore absorption, the green color, should overlap with the another fluorophore which is targeted to the acceptor. The green is donor, the red is acceptor. If you excite this green only, and the intensity of the donor decreases or the lifetime decreases, the sensitivity of the increase in intensity in the acceptor channel, meaning that it is called as a sensitized emission. Some people measure the lifetime of the acceptor too during this condition, but mainly we follow the lifetime of the donor in the situation of the FLIRR. This will happen only when the distance is within 10 nanometer between donor and acceptor molecule and the dipole moment. Dipole moment is FLIRR is also called as a distance dependent, and the dipole moment is very important. The donor absorption dipole moment, donor emission dipole moment should variate with acceptor dipole moment. It should be 1 to 4. If it is perpendicular to each other, then there is no FLIRR. So this is a very important parameter, the dipole moment. Even though if you select appropriate fluorophore which overlaps the donor and acceptor, it should have the distance of the molecule. When you choose the protein molecule to investigate, you should know the distance between the two molecules is close to 10 nanometer, and the dipole moment is another story to see whether the donor dipole can find a partner in the acceptor absorption dipole. That's a very important parameter to achieve the FLIRR signal. So this is the R0 value. Foster derived an equation where you can see the kappa square and involved, and this value is orientation, or we call it as a orientation factor, and 0 is the dipole, and kappa square at the maximum is 4, and the dipoles are in series. Any kappa square between 1 and 4 will be okay for the FLIRR. So what's the dipole moment? It's electromagnetic radiation, as many of the if you know the physics of it. If we have the donor and acceptor, if it is parallel to each other, you get FLIRR. If it is collinear, you get FLIRR. And if it is an angle, you will also get FLIRR. But if it is perpendicular to each other, the blue color is this way, and the another dipole, it's at the center of the dipole is there, and you will not get any FLIRR. But if you have in a situation in biology where there is not a one-to-one study, there will be a more molecules surrounded by the donor. In this kind of situation, you may be measuring average FLIRR. That's why we say the FLIRR efficiency is not absolute, it is approximate estimation of the energy transfer efficiency. So here is an example to show you. It is RAC1 and IQ-GAP1 targeted with the EGFP and M orange, and you see the co-localization. Green color, you see the distribution of the donor and acceptor in a spectral FLIRR imaging. If you put the cursor on the yellowish color where it is red and green co-localized, you see a sensitized emission, the decrease in intensity of the donor, increase in intensity of the acceptor. You see the contamination of the donor bleed through and other contamination involved. That has to be removed if you wanted to quantitate the signal of the FLIRR. That is where we developed a software package where you can just load the images, about 7 images. You can able to get the correct information, remove all the out of focus signal, all the the bleed through signal, and as well as when you excite the donor, you may be exciting the acceptor too. So we change the equation of, if you see any textbook, the energy transfer efficiency is calculated, intensity of the donor in the presence of acceptor and in the absence of acceptor. If you take the lifetime, it is in the lifetime value of the donor in the presence of acceptor and in the absence of acceptor. So if you use the intensity-based method and compare with the lifetime, it is almost close if you apply the software to remove the crosstalk or contamination involved in the measurement of energy transfer efficiency. If you do not want to calculate or estimate or quantitate the signal, you can just show FLIRR occur or not. You do not have to do this, but you have to worry about the crosstalk of donor to the acceptor because of the spectral overlap. This is what mainly we train in our workshop, how to optimize your imaging condition and how to do the data analysis from the microscopic images. There are a number of technique approach you can use. The same software can be used. Lifetime is a simple method which you need donor and acceptor, but you have to do all kinds of calculation to interpret your biological events. So here it is, I am showing 2-color FLIRR, confocal FLIRR, by using quantitative signal. E person is in the y-axis, x-axis is acceptor to unquenched donor ratio. You can able to interpret the data by correlating these E person versus the correlation acceptor to donor ratio. And you can also do 3-color FLIRR, 3-molecular FLIRR. Here is the NWASP and IQ-GAP1 and CDC42. In the absence of CDC42, you can see very good FLIRR signal between NWASP and IQ-GAP1, whereas in the presence of CDC42, the FLIRR signal decreases. The reason could be there is a distant increase between NWASP and IQ-GAP1. So now I will go back with this principle of using this tryptophan and a bound NADH, because there all the FLIRR work you have to target with the fluorophore. Here there is no fluorophore is involved in measuring the NADH and the tryptophan, because these are all auto-fluorescence. As you know, the tryptophan is a neurodegeneration, cancer, and immune regulation. So when I got into the field, as Wolfgang mentioned, in 2012, when I read an article about the tryptophan, it decreases by biochemical method in the cancer. So I thought I should work with this tryptophan work rather than doing NADH, but of course we finally ended up doing NADH work. But it's a challenging one. Imaging tryptophan is not that easy because UV, the microscopy provide you 400 to 700 nanometer transmission of the microscope optics. So we have to reconfigure our BioRad 2002 radiance at the time I used, and we reconfigure the microscope so that I can collect directly from the using on the non-descanning method. We were able to detect the tryptophan signal. So it provides you metabolism, tryptophan metabolism will provide you number of age-related pathology and lifespan. So it's an essential amino acid, precursor of niacin, which in turn is precursor for NADH. The increased tryptophan catabolism and increased interolamine dioxygenase activity in the kynurenine pathway are linked to cancer development and progression. So tryptophan investigation, our cancer center people are very much interested in the kynurenine pathway to investigate the tryptophan. So phosphorescence energy transfer will allow us to investigate the mechanism of degradation of tryptophan, and combining this FLIRR and FLIM may be able to provide the information about the cancer progression using the tryptophan method. Its clinical relevance includes pathology in cancer, neurodegenerative, degenerative diseases. It's also used for marker for the level of protein abundance. So how FLIRR occurs between tryptophan and bound NADH? We hypothesize that NADH is interacting enzymes carrying tryptophan residues. So you can see number of residues carries carrying the tryptophan residue responsible for quenching the tryptophan lifetime. So that could be the reason why we are seeing the decrease in lifetime of the tryptophan in the presence of bound NADH in the protein. So I clearly see we use the same idea of prostate cancer cell with the doxorubicin, and you can clearly see that increase in energy transfer efficiency with the time. This is a control, no drug, and the same way for African American cancer cell, you can see very clear increase in the redox ratio versus the E%, tryptophan E%. And so many questions came when we submitted paper that the doxorubicin, if you keep it for a long time, it is apoptosis takes place. It may kill the cells. So we thought that we should use that approach to demonstrate what we are seeing, the decrease in lifetime in the tryptophan is mainly due to the residues, the coenzyme carries, quenches the lifetime. So we did the apoptosis work. You can see after 12 hours, you can see the apoptosis occur with the doxorubicin, 12 hours and 24 hours. We use the link cap on both cells, but all our experimental data collection is in seconds, not hours. So we are not saying if you use 1 minute, you saw in the previous slide, I showed you 60 minutes, the morphology changes with the doxorubicin. So naturally something is damage occurs to the cell with the doxorubicin drug. So we wanted to use that and test whether the FLIRR occurrence is true or not. So here it is, after 24 hours, you can see the morphology of the link capsule is completely gone. And you can see this is the control. There is no increase in energy transfer efficiency, same as in the African American cancer cell line. So what we observed in the change in decrease in lifetime is mainly due to the bound coenzymes of NADH. We also tested the cervical cancer cell, HeLa cell. Here is the bottom is the control, and you can see increase in energy transfer efficiency in the tryptophan and NADH interaction in the cervical cancer cell too. The same doxorubicin we used to test the cervical cancer cells. And I just show you another few slides, I am done. Monitoring metabolic changes in non-adherent leukemia cells. Non-adherent means it will not stick to the cover slips. So if it is not sticking to the cover slip, we cannot image it. So we went ahead and figure out a microfluidics array. So it will also has the position marker You can track the position. It also traps the cells. So that means you can add the drug, keep it in the incubator and bring it back, and you can follow the position and again study with the time how the drug response takes place in the leukemia cancer cells. We use the AML cell, acute myeloid leukemia, and the drug we use is ceramide nanoliposomes. And here we see that the drug response, it shows after 12 hours, only you see increase in the coenzymes, NADH bound coenzymes, you can increase in a signal which we use the lifetime here. Of course, all the approach is the same FLIRR approach we use to calculate, estimate that. So here you see that number of cells are there each well. We would like to go for a single cell. So we are currently working on how we can improve the microbial array to trap only a single cell so that we can able to see the response of the drug from the single cell. All right, we are collecting a large number of data. As I mentioned before, it takes a longer time, a week to calculate all the region of interest calculation, et cetera, et cetera. So we thought that we should use the machine learning approach. There are a number of ways you can do that, unsupervised learning versus supervised learning. So we use the machine learning approach and to compare the data. So here it is. So you can see clearly, this is our FLIRR, 2 parameters used. This is the machine learning approach. 8 parameters we used, all the pre-exponential factor, lifetime values, etc. So you can see the response, the yellowish color, within 30 minutes, you can see the drug response. We don't have to wait for 60 minutes. What it does is that you can quickly see the response within 30 minutes. What we did after fitting the lifetime data, you take all the parameters and feed it to this machine learning algorithm, and you can see that it takes only maybe seconds to minutes as compared to the 2 parameter FLIRR value we have been calculating using our image segmentation software, maybe 1 hour or maybe something like that. It takes more time. More importantly, you can get it within quickly 30 minutes drug treatment instead of 60 minutes drug treatment. You see that 60-minute drug treatment, the cell morphology changes. So this helps a lot. We are working on this right now. All right, so I showed you the FLIRR approach using the lifetime imaging microscopy instead of intensity. Both method is good, but it gives sensitivity, particularly in the case of tissue. We have also demonstrated the FLIM-based REDOX ratio advantages in clinical imaging too. We have also demonstrated FLIM-FLIT interaction between tryptophan and bound NADH. FLIRR approach we use for prostate, cervical, and leukemia cancer cells. We are also working on mitochondrial dysfunction on Alzheimer's disease. The non-invasive FLIRR assay and the machine learning can be used to investigate or monitor mitochondrial dysfunction in many diseases. Thank you. Stay safe and healthy. The next meeting, we had a 30 years of multi-photon microscopy last January. The next meeting will be next March, next year, 2021. The abstract due date is August 26. So please spread the word and ask them to submit the abstract. Thank you very much.